Guys, focus. Come on. I got stuff I want to cover here. Come on.
It's loading. It's not loading.
Okay, so we've had at least this one. Is it H-E-N or H-E-M-E? Is it okay? I saw it this morning and saw H-E-M-E. Like H-E-M, and I was like, I think there's one on there. All right, so it's heme, but it is only H-E, not H-E-M. But okay, let's get some examples.
Hemorrhoid. Hemorrhoid.
That one is spelled really weird, but it does have to do with blood. So I'll give you that one. But what we're going to talk about is combining these two. Okay, we had fill or file back with hydrophobic and hydrophilic, right? And it's been a long time. But when you combine those, you can get a disease where somebody can't stop bleeding. Hemophilia. And it's like, if you define it, it means like to like blood, but it means basically you get too much blood that is leaking out of the body. Hemophilia is an example of non-Mendelian genetics because it's on the X chromosome. It's on one of the sex chromosomes. And that means gender has a lot to do with who gets it. And we're going to talk about that. Color blindness is the same way, so I'll wait until we get into that and explain kind of what's going on with it. But that is an example of a disease that is non-Mendelian. Polysaccharide, we've had that one a long, long time ago. So polysaccharide meaning made up of many glucose molecules, right? A really big starch molecule is described as a polysaccharide. But we're going to talk about genes, or maybe you have multiple genes giving you one trait. We call them polygenetic traits. And that means that you would have to do like I don't know, if you have 5 genes controlling maybe like height or something like that, then you'd have to do all 5 Punnett squares to see what the combination was. And so they can be, in fact, most things that are traits of humans are called polygenetic traits.
Hemisphere.
Hemisphere, half of the planet or half of a square or half of a circle, I'm sorry. And we're going to talk about things like hemophilia is an example that are called, spell it right, Z-Y-G-O-U-S, hemizygous. And last week we had homozygous and we had heterozygous. And hemizygous is an interesting one because if we have, let's see, I'm going to do this up here on the side just so we can all see it. Like big A, homozygous dominant, big A, little a, heterozygous. But what if we had something like an X chromosome? And then on the X chromosome, we have the gene for hemophilia, right? But then that person has a Y chromosome, which does not even have that gene on it. Well, they only have one copy, so we can't call it homozygous dominant because they only got one. We can't call it heterozygous because they only got one. So we call that hemizygous. And we put the little superscript up there to say that it's on the X chromosome. Co-adaptation, as a result of like co-evolution. So you could think of co-adaptation as being like flower colors and then bees of like appreciation of, you know, like art or anything like that, but bees liking flower colors, right? That's called co-evolution and co-adaptation. We are going to talk about traits known as codominance. When you have more than one dominant allele, say like blood types, right? You have type A blood, type B blood, but then you also have type AB blood. And so we're going to talk about how that happens as like a non-Mendelian example. And in this case, I believe it means not.
I don't know. I know where to throw in. I was going to say internal, but I don't know.
So internal would be the prefix inter. You're right, it does start with in, but it's the prefix inter, which means inside. So
Yeah.
that one's not going to help because like it's, you know, that kind of fuzzy area where the letters are right, but it doesn't have that meaning. Incompatible, to not be compatible. Two people are fighting all the time, they would be described as incompatible. We are going to talk about incomplete dominance when one trait is not dominant over the other one, neither one is dominant, and we can all of a sudden get pink flowers, white flowers, and we'll see some traits blend together like that. So clearly still talking about genetics and still doing Punnett squares, but trying to toss some wrinkles in because hopefully after the horned monster activity, we all understand how to do basic Punnett squares. If you're still a little fuzzy on the big diagonal cross Punnett squares, I have one on the assignment today, so you'll be able to do one of those and I can kind of walk you through that. I fully expect that. That's normal. So, any questions on this? Then let's go to this. Non-Mendelian genetics. Okay, so put your computers away, and we will go over this. I don't think it's going to take us all that long.
You still have mine. Bring it.
Okay, so on the bottom of this it says, guys, can we move on? Why is it happening? Clearly. All right, so at the bottom it says it's not all just green and yellow peas. So the point of this is to say it does get more complicated. And at the top I've got pictures here. And so this on the top left is showing a really large Punnett square. So you never have to do one of these. And that would be a 64 square Punnett square, I think. Yeah, it is. And it's, so what it's showing is polygenetic traits and how skin color is an example of this, depending on these three different alleles, and skin color in itself is more complicated than this. But you can get a range of different options. So it's not just like two different skin colors. It is a spectrum of differences, right? Height is the same way. Eye color is the same way. And so we'll talk about polygenetic traits. We'll probably start off with this one, if I remember correctly, because it's kind of the simplest one, where you do get this blend of traits. You can get red and white flowers crossing to get pink flowers. Codominance is blood types right here. And I'll explain maybe a little bit about this. Like, I don't know this cat. I've never met this cat, but I know it's a girl. Because that color pattern, anybody know what it's called? Calico. That's a calico cat. It's just a name describing the color pattern. Is impossible to get in boys because that color gene for the hair is on the X chromosome. Well, how many X chromosomes does a boy get? One, because the other one's a Y, right? So they can't even get a blue and an orange allele because they only get one from them. Now, you can have a white and black or white and orange, but you can't have white, black, and orange. It just doesn't work. So this is all stuff that Mendel didn't know about, probably because, it was the 1800s and he was a monk growing peas. But the first one is called incomplete dominance, okay? In this case, the heterozygote is going to be a blend of two alleles. And so our example for this one, and this is actually how a lot of them work, is flowers. Because if you take a red flower, really bad drawing quick flowers here, and cross them together with a white flower, you are going to get a pink flower. So what letter should we use for the red flowers? R. I agree, right, and that kind of makes sense. But then what letter should we use for the white flowers? And that's where we're not going to use a lowercase r, right? A lowercase r would represent like a recessive allele, but there is no recessive allele. So we can do this and then realize without even doing a Punnett square, right, that this RW is pink. So the heterozygote is a blend. These colors seem to blend. So what happens if I breed two pink flowers together? What do I get? If the colors blend and I blend pink with pink, then what do I get? I get pink. So that's not what's going to happen. You have to think about the Punnett square here, where I have RW cross to RW. RW, RW, like that. What am I going to get? I'm going to get all three colors, right? I'm going to get red, 25%. I'm going to get white, 25%. And I'm going to get pink, 50% of the time. This is really different than what Mendel saw, right? If this was Mendelian, if this was like the peas with the greens and the yellows, then this would be red. And if we had red cross to red, we would get 75% red and 25% white. This is not the same, right? But it still works with Punnett squares. The reason people miss this is they start thinking the colors are always blending. Well, I'm going to breed pink flowers with red flowers, and I'm going to get pinkish red flowers. And I can breed those pinkish red flowers with red flowers, and it'll get a little bit more red. And it doesn't work like that. It's just that the heterozygote is a blend of the two alleles, right? The trick is, the question will have to say, okay? It has to say, in this example of incomplete dominance. Or it'll say, in this Mendelian trait or something like that. And so you just got to pay attention to what the question says, because it will specify kind of, which type it is. What about this? What about codominance? Co means with and together, right? And so one dominant trait with another dominant trait. You can choose whatever you choose. I'm not going to find it. But it's your choice. It's your choice. Entirely your choice. I'm saying I would. Okay, so we got two dominant alleles, but we still get a recessive allele, okay? And this is blood types. Anytime you see the superscript letter on top of another one, that means that they're one thing. Okay, so this is a blood type allele that gives someone that blood type. I'm actually not going to explain a ton of like why we can't get certain blood types, because that's immune system stuff. And these are the other two. The I stands for immunoglobulin. It's a little thing on the surface of red blood cells that either like your body recognizes and doesn't care about, or it doesn't recognize and it tries to attack and destroy it as like a pathogen. Yeah, right. So if these are the alleles, what are the different genotypes? What are the different combinations of two of these that I can get? You got to combine two of these, IA, IB, and little I. Give me a combination. IA, IB. Okay? That's the same thing. Remember, like, big H, little h is the same thing as little h, big H. We just don't write it as little h, big H. So I'm not going to write that one down. That's the same thing. IA, little I. Got it? Which one? IB, little I. We got three more. You're avoiding the homozygous one. Oh. There we go. IB. Yeah, there we go. Okay, so these are, from 3 different alleles, we can get 6 different genotypes, and that will give us, so we can go in this kind of pattern, the phenotypes. What blood type do we get from this one? Or from these two, or from these two, or from this one. You'll know your 4 blood types. We don't care about positive or negative. That's a completely different system that we're not really concerned about. If somebody gets the genotype of IAIB, what blood type will they have? Well, here's the 4 different blood types, right? I put them up here, and I actually don't know how much you guys would have learned. You've got A, B, AB, and O. that's, so we've got blood type A, B right there. What about underneath here? A. And what about right here? B. And then what about right here? That's O. Okay. So if all we had was A, B, and O, well, that would really not be codominance, right? But the fact that we have this blood type right here that is a little bit of both is actually, that's the evidence of codominance, right? Because we have two dominant alleles right here. Just as a quick little thing to see who knows, like, what blood type would someone want to be? And this is all about receiving blood in a blood transfusion. What's the best type of blood to be? Which one? AB. Because if you're AB, then you can get O blood. You can always, of course, get your own type of blood. You can get A and you can get B because your body recognizes all of those types of blood as a part of you. And so you're good, right? Like you can receive any type of blood. What type of blood do you want other people to be? O. Because O is the universal donor. Those blood cells don't have the little signal on them at all. And so type O blood goes to this one, to this one, to this one, and of course to yourself. So it's really good to have type O, like other people have type O blood, whereas you want to be type AB blood. And I don't want to go into exactly why, because the point of this is to say the genetics of it is codominance. And maybe we return to it as a little review next week or at the end of the year after the sergeant, something like that. But there's two dominant alleles right here. There are other examples of this, right? So let's say that you have a chicken, and you have a black chicken and a white chicken, right? And you breed them together. If you get a gray chicken, is that going to be codominance or incomplete dominance? It's not, because gray is a blend of the two, right? Whereas if you happen to breed this black chicken and this white chicken together and you get a speckled chicken where some parts of it are black and some parts of it are white, then that would be codominance. And I'm pretty sure that's what certain chicken patterns do. So you have a black chicken, you got a white chicken, right? And when you breed those two together, you'll get both of them, and you can see both the black and the white feathers. And this is the real, not a very good picture, but there's the black and white chicken. The traits aren't blending because it's both black and white at the same time. So it's not incomplete dominance. That one is an example of codominance. So. All right, sex-linked traits. Can everybody see the picture? Move it over here. You all see the picture? You see what it is? Now, if you're colorblind, you can't see what it is because that's called red-green color blindness, and the eyes cannot distinguish red versus green, right? And so that means all of these dots up here is the same kind of red-green blend, which is a little bit brownish, and so you can't see these kind of things. This is a test for color blindness. A lot of times at the doctor's office for little kids, they would have like numbers in them or something like that. But we use these. I use them just as an explanation of like this is an example of incomplete, not incomplete dominance, of non-Mendelian genetics, because I've had plenty of students that are colorblind, right? Like every year, and probably I would assume this year I would have one, but roughly on average one a year I have a student who's colorblind, but they've all been boys, right? Like it's possible for a girl to be colorblind, but it's incredibly rare, and we can see that if we look at the alleles giving us the genotypes, giving us the phenotypes right here. So the alleles for this are, and it's called a sex-linked trait. It's on the X chromosome, which is called what it's called the sex chromosomes, right? So it's sex-linked trait on the X chromosome. The alleles are X, big R, X, little r, and Y. So just like that IAIB, you cannot separate the X big R, the X and the big R, right? That means it is a thing on the X chromosome. So now we need genotypes again, right? What are my combinations of 2 of each of these? What's it going to be? You got to combine these. Give me 2 because we got a mom and a dad. And we'll put those right here, just like we did with the blood test. You got to say bigger, little. X big R, X big R. Good. That's one. X little r. That's another one. We got 2 more. X big R Y. Oh, that's weird. And last one. X little r y. Okay, and then whenever you're given a question on this, you have to think, okay, what's the gender and what is the disease? What is it there? And we would consider this a disease because it negatively impacts someone's life, even though it's just like in choosing clothing that matches or knowing which one of the lights is on a street light. And so X big R, where girl? Girl. X is girl. So this is a girl. And this is a sex-linked recessive disease. So this is the bad one, right? This is the one that can't see colors. So we would say girl, normal. Well, then what's this one? Girl, colorblind. I'll just do that. Colorblind. Unnormal not. Is that what you said? Unnormal. We can call it, let's call it colorblind. What's the next one? Girl normal. However, I do want to put a word on here. She's a carrier. A carrier is another word for a heterozygote. They have the allele, but they don't have the disease. But that means they can pass it on to their offspring. What about this one? Boy normal. Boy colorblind. There is no boy carrier because the boy doesn't have another X chromosome. These are the 2 that would be called hemizygotes. And they're called that because they only have half of the number of alleles as the girls do. Now, it's perfectly fine. We, as boys, can get colorblind, or can see colors with only 1 copy of that gene. But why is it so much more common for me to see boys that are colorblind than girls that are colorblind? I don't want to say it's 50-50. In certain cases, it would be, but it's not always, right? How many bad copies does a boy need to be colorblind? Just 1, right? Because they've only got 1 X chromosome. A girl needs 2 bad copies in order to be colorblind. And that can be kind of tricky to happen, right? Because you have to have a dad that is colorblind and a mom that is at least a carrier. Whereas with boys, all you need is 1 bad copy. So if a boy is colorblind, and this is a good time to introduce the idea of pedigrees, which I'm not going to do in great detail, but if you have this, dad, mom, boy, that's a pedigree, right? Square, and we'll go over this before the test. That's kind of my idea for Thursday is to do pedigrees and then take the test. If boy is colorblind, we would fill them in. Whose fault is it? Was it mom or dad's fault that boy is colorblind? Every class always says that, but think about what it looks like. X little r y. What chromosome did the dad give the boy? Because he's a boy, right? So X, Y, maybe dad is even colorblind, right? That's irrelevant because dad had to pass on the Y chromosome. So it turns out that this this must have come from maybe mom was a carrier, right? So any boy sex-linked trait on the X chromosome is passed down by the mom. This is exactly what happens with hemophilia. And it happened with hemophilia, and there are these huge pedigrees that I'm going to show you real quick. Let me see if I have it up ready to go, and then we'll come back to it. Okay. Giant pedigree. I've kind of made it as big as I can. And it's a famous pedigree. You may recognize anyone from like history class or something.
I can't.
Need glasses.
I'm wearing glasses.
You need better glasses.
Names are small.
There. You know, read it. That's Queen Victoria. She was a very famous queen of England, right? And she ruled, or lived at least, I would assume she ruled until her death, which was a very long time. 1819 through 1901 is when Queen Victoria lived and ruled England, right? So she started this, she didn't start the family, but she had lots of kids. And in fact, like you can kind of tell, how many kids did she have? 1, 2, 3, 4, 5, 6, 7. What? What's wrong?
Why they wait?
It's a little weird, right? Royal. Okay, there's two things here. One, I think that's a mistake. No, it happens a lot, but not with children. And the reason I can tell you, look, it happens again right here, right? With Queen Elizabeth, who married Prince Philip, who just died a week ago, right? And they were cousins because they can both be connected right here. Yeah, they were. Queen Elizabeth and Prince Philip were cousins, right? But it's that whole like royal thing. I agree, it's weird, right? I'll show you another one in a second that's from the Spanish royal family that has a lot more. But I don't think that that's actually true. I think that one is a mistake. That's Alice and Louis of Hesse. I do believe that one is a mistake. So they may also have been like cousins or something like that. But that's not the reason I put this one up. One, it's just interesting to look at like royal family trees where they do blend together because the current queen of England had married her cousin, but I think it was more like a second cousin, right? But the reason I put this up here is because it shows hemophilia in the royal family, starting off with Queen Victoria, right? Because every single person that is fully shaded in was a boy that has hemophilia. Do you see any girls that have hemophilia? It's possible to have a girl that has hemophilia. It's incredibly rare, and especially back then it would be super rare, because do you see any hemophilia boys that had children? One. Because what happened to all of the rest of them before they had children? Their blood wouldn't clot. What would happen? They died. They wouldn't live to childbearing age, right? Because you fall down, you get a scrape, or you get a bruise, and it bleeds inside. And so Leopold was the only one that lived long enough to have children, and they had, it looks like, 2 children right there. There's a lot of really weird things that are going on right here. Like this one right here, this was in the early 1900s, and this was the Tsar of Russia. And this was, if you've ever seen the movie like Anastasia, she was the, have you ever seen the animated movie Anastasia with that flapping talking back thing? It came out when I was a little kid. But there was this rumor and myth. This entire family right here was murdered during the revolution in Russia, where the royal family was killed when communism rose in Russia. So there's all these kinds of connections to other kind of topics and ideas. And in fact, like today we have Prince Charles, Prince Andrew, and Prince Edward. The current, royal family of England is right here. So all these, royal families kind of, they're all connected a little bit. We can see Russian royal family, Spanish royal family, and the English royal family all right here. But hemophilia was a big deal as far as like this family goes. This was the Spanish one. What is their relationship aside from having kids together, having Anne of Austria? I think they're cousins. So you go up here and you see that Charles and Ferdinand were brothers, and so that makes them first cousins. These two are second cousins. This who is Charles to Maria? Not dad, huh? Uncle. These two are also cousins. These two are like third cousins because you can connect them, but you have to go all the way back up to like right here. And then once again, we've got Mariana of Austria, and that would be an uncle right there. And by the way, this Charles of Spain was the last of this royal family because he had a lot of genetic problems, right? And everybody knows that people shouldn't marry, have kids within their family. The reason is all of these people have similar bad alleles, right? There's a whole bunch of things like big A, little a, big A, little a, big A, little a. And other families are going to have different bad alleles, but not the same ones, right? And so the problem with a family doing this is that you start multiplying the effect of bad alleles. And it's like breeding dogs, right? If you breed all the dogs together, all the dogs together, all the dogs together, you get very similar looking dogs. But then all your labs get hip dysplasia. And so it's kind of the exact same thing. But he had a lot of genetic issues right there. So, all right, those are pedigrees. We'll look at pedigrees in more detail. But I do need to finish this. That explains what we call sex-linked traits. And our last one is polygenetic traits. My example was skin color. Because if you think about skin color, and there's two different ways we could graph this, right? If we have the number of people, or we could say the percent, and we get something like the lightest, the medium, and the darkest. Light, medium, dark, and the number. What we do not see with skin color is something like that, right? That's not what it would look like if you graph the number of people or the percent of people that have a certain skin tone. What you are going to see is a shape of a graph that looks like that. Because there's thousands of different possible combinations of different alleles, right? And it would be maybe rare to get all of one of them, or rare to get all of the other ones, but it would be pretty common to get a mixture of all of them. And so most people fall somewhere in the middle of this. Of course, this depends on like culture and where you're from and all kinds of stuff. But in the absence of all of that, we would see a spectrum like this of all people. This works with height, right? If you take males, are most males going to be 6'8"? Are most males going to be 5'2"? Most males will fall in that range of maybe like 5'8" to 6 foot to 6'1" or something like that, right? But occasionally you will get somebody way up here and somebody way down here. And that's how you can tell that it's a polygenetic trait, because you see a range of different traits. And it turns out that this is how most of our complex traits look. The trick with non-Mendelian genetics is thinking about how it's different. How are polygenetic traits different than Mendel said? How is sex-linked traits different than what Mendel said? How is codominance different than what Mendel said? And how is incomplete dominance different than what Mendel said? And the best way of kind of understanding that is by doing some practice. So that's the assignment thing. Let's look at Punnett square practice. Put it on this morning. Hopefully everything works. There it is. Remember, this includes Mendelian and non-Mendelian, so you have to read the directions on each little section of it. So tomorrow, or not tomorrow, but Thursday before the test, we will do more pedigree practice. We'll have question sets with pedigrees, and then we'll take the test, and that will kind of blend in a little bit of everything. All right, so this one says complete the following Punnett square crosses and answer the associated questions. The question sets include monohybrid crosses, a dihybrid cross, and some non-Mendelian questions, which is why it's kind of a combination of everything. Any problem in yellow has been completed for you to help with the others. Any square in green has to be filled in, and that includes Punnett squares. And so you'll see the Punnett squares are just little tables that are shaped like a Punnett square, right? And just to remind you, genotypes are just the alleles. Phenotypes are the physical traits. So if a question asks you like what percent are going to have this phenotype, then that would be like a trait, right? The capital letter indicates the dominant allele. The lowercase is the recessive, unless of course it's incomplete dominant. It's too capital. Gamete alleles and dihybrid cross come from an independent assortment. So big A, big B, little b will produce the sperm cells of big A, big B, big A, little b, big B, and little a, little b. That's what we did last week, and that's the part that most people struggle with when we get to dihybrid cross. Traits blend in incomplete dominance. Both traits are seen in codominance. Sex-linked traits are found in the X chromosome, but are more common in males. They only need one copy since there's only one X chromosome. And we don't do polygenetic traits because the Punnett squares get huge. We just understand that it would not be a Punnett square, a simple one, because it's a blend of everything. So, all right, and then I've got a simple Punnett square here, and this one is a tall plant cross with a tall plant, a tall plant cross with a short plant, and then we get to the dihybrid crosses where I filled this one in for you, and I answered the question. And then you get to your dihybrid cross, so there are the organisms that are going to get crossed right there. You fill that in, and then I've got 3 questions. And then we get to the non-Mendelian. So I've got a codominant question. I have got looks like I have 2 codominant ones instead of an incomplete dominant one. Maybe I made a mistake right there, but that's okay. A sex-linked trait one with hemophilia. A sex-linked trait with fruit fly eye color, which is how that works. Well, there's incomplete dominance right there. And then there is SpongeBob and Sandy, and it looks like this one is an incomplete dominance because it's red and blue crossed to make purple. It's a little bit of everything, and if you don't try and figure out what you don't know, then you're not going to know which questions to ask. So let's just get started, and we have a long time for this. Let's see how much we can get done.